Well, hey, Biology 11. This is our final topic in Chapter 20. It's 20-5, the fungus-like protists. It's one of the more interesting and sort of cryptobiology-like topics that we go over. You have to realize that in this chapter, you've been studying something which is, by and large, the kingdom of the single cells. They're eukaryotic, and they seem to be a little confused. Some are plant-like, some are animal-like, some are fungus-like, but they don't have the characteristics to put them in those other kingdoms. These are the interesting sort of unicellular lookalikes, a lookalike kingdom, if you will. So we have to finish off this topic with our fungus-like protists. We'll get going with that. A quick tour of the course site shows you Kingdom Protista, and right here, there you go, Lesson 4, Fungus-like Protists. What is in that lesson? What should you take in? Well, I've got a little bit of it hiding out here. Let's see if I can call that up behind. Oh, what the heck? Let's just click on it. Get there faster. Okay. Now, there's a few animations that you'll see here. This one, for example, tries to start right away, so I'm going to pause it. Otherwise, it'll just sort of take over.
A few unrelated groups.
There we go. Looks like I'll have to pause it again. Oh, well, not a problem. There we go. So what we have on here is I'll be posting the video to this position. Your notes will be here. We've got some videos for you, a couple of neat animations on things called cellular and acellular slime molds, discussing what they are and how they're a little bit different, and a nice picture of what we call a slime mold known as dog's barf. I love the name so much I had to put it on the site. Nice, brilliant pink. At first glance, somebody would think something like this would be a fungus, but it's not. It's a group of protists that are acting in a group way as if they are a fungus, but they're not kingdom fungi. I'll make a point about kingdom fungi before we move on. The only way you get to be in kingdom Animalia, kingdom Plantae, or kingdom fungi is you have to be multicellular, and you need to be multicellular pretty well all of the time. Here's another one about kingdom fungi. They have cell walls made of chitin. It's an interesting material you find in the shells of crabs and crustaceans like shrimp and lobster. Now, fungi use that to help them stay rigid and stand up on land. You don't ever see that in fungal-like protists. So these are little mimics. They might group up like a fungi, but trickery, they're not kingdom fungi. So let's begin with a neat little sort of peek into 70 years of research into slime molds. I'll show you this one. Slime molds are fascinating. Let's let that come in. Oop, pause. I want to... Let's go full screen. Okay, check this out. Now, this little thing moving around is basically a group, a conglomerate of little slimy amoeba cells, and they've come together as a group and they can move around. They call this a slug. Now, John Bonner has worked with slime molds for a very long time, and the way they interact and the way they behave after his years of research with slime molds, he claims it's almost as if they have a group intellect. They're fascinating. Check this out. Pause. Okay, stop the pretty music for a moment. What you're seeing are individual amoeboid cells. So they're just like the amoebas we've looked at before, but in slime molds, they love to colonize, and these little amoebas will come together. They'll touch their cell walls, and they'll act like sort of a large conglomerate, basically as if they're one organism. Watch them come together. It's amazing. let's pause it for a second. You can see them coming together there. They're very colonial. The next distinction is that they can come together and parts of the conglomerate will differentiate. Now, we haven't seen that. You and I have differentiated cells. We've got nerve cells up in our cephal region, up in our brain, right, up in our head, and we have specialized cells for our heart and for our digestive system. But when you think about protists, there's not supposed to really be cell specialization, right? Well, not necessarily. When these cells come together, they decide to form almost like a mushroom cap of sorts. And the cells at the bottom hold up the cells on top. And it's the cells on top that release spores, and they differentiate from the other cells. Those poor little cells at the bottom exhibit self-sacrifice because they die, and it's part of the overall life cycle. Now, this fruiting body is just more or less, if you think of a mushroom cap, you're not far off. It's going through a reproductive phase here. Now watch the rest. Great little video there. I think it's a fabulous introduction to our friends, the protists. So slime molds. We'll come back to some of the animations on the site, but for now, we'll go through our course notes, and I'll give you a chance to fill in what you need to know. So let me turn on reflection here, and there we go. We're golden. So just like fungi, what a fungal-like protist would do would be to be a decomposer. In fact, that's the main role of anything that's fungus-like. Now, if you've ever had athlete's foot, there are types of fungi that do act in a parasitic manner. And wait till we get to kingdom fungi. I've got a great multicellular fungus to show you called cordyceps, also known as the zombie fungus. But that's for later. For now, what I want you to know is that the protists that we're looking at right now are fungus-like, but they are not fungi. And here's the reason. I don't need red, I need purple. The protists that we're looking at are not multicellular natively. They don't have cell walls composed of chitin, which you would see in kingdom fungi proper, but they do act like them. So because they don't have these key aspects to them, you could say they're fungus-like, but they're not really fungal. Not to worry. There's plenty to learn. Now, it's interesting how there's a point being raised here that they're heterotrophs, so they eat other things, and they absorb nutrients from dead or decaying organic matter. Well, we've seen a term in this course for that. Dead or decaying matter that you eat. Well, let's call it for what it is. That means that they are saprophytes. And the way I help students to understand this is, if you've ever seen the movie Jaws, okay, Jaws is a heterotroph. He goes out, hunts down a meal, and eats it. It would be a little bit different if Jaws was going around finding only dead material to consume, right? That would be saprophytic type living. So there's a couple of interesting points being made down here. Most true fungi have chitin in their cell walls. Okay, we get that. There's I think it's a bit odd right now, unless you've done a lot of cellular biology. Fungus like protists contain centrioles. Now, centrioles are something you see really only in animal-like cells. So what are they doing in something that's fungus-like? For the longest time, we thought fungi were just some odd type of plant. Plants never have centrioles. So when we look at something that's fungus-like, we never expected it to have centrioles in the first place. Centrioles are a very non-fungal little piece of the cell to find. It's an odd sort of reference, unless you've had a lot of cellular biology, but when you see centrioles, those are things that help animal cells divide. Strange to find them in something that's sort of fungus plant-like, right? So the centrioles are a little odd to see. I would focus much more in on the chitinous cell walls that you would expect to see in fungi. Defining characteristics of the slime molds are coming right up. So we already knew that they were fungus-like, and they're really good at recycling decayed organic material. Gotcha. It becomes sort of vital to look at their life cycle because it turns out there's two varieties of slime molds here. One where the little cells start out as individuals and they can group up, and then there's another type where they pretty well always group up. They don't even hang out as individuals. That's the two main varieties that we'll look at here. And this is what I wanted to get to because I thought the previous slide didn't really do it justice. So, type 1, we've got the cellular slime molds, and type 2, acellular. Let's go back a little bit. A is a Latin prefix that means without or not. And what they're meaning to tell you here is when they start their life cycle, do they start out as individual cells or not? The acellular slime molds do not, whereas the cellular slime molds are cellular at the beginning. They just choose to group up. So I'm simplifying, but you'll see why in a minute. Zoom here. All right, so in type 1, the cellular slime molds, you can think of it as a giant group of amoebas, and they've all come together in a giant amoeba bag, a big slimy sheath. The acellular slime molds, it's like they're not even individuals anymore. Their cells have fused, and all the nuclei from all the cells have crammed into the middle. It's like a giant supercell. The cellular slime molds are like trapped individuals, but the acellular slime molds, they've merged. They're just part of a larger structure now. And it's very odd. At this point, you sort of look at it and you say, oh, are acellular slime molds multicellular? Not really. It's like a giant mix. And we'll get into the life cycle here in a moment. So, at this point, I'll mention something about the cellular slime molds, then I'll jump out to an animation of these to sort of help you out a little bit. The cellular slime molds begin as free-living cells. That's a huge point. They are individuals. You'll find them in the soil, and they basically look like soil amoebas. When times are pretty good, they'll go through a lot of asexual reproduction, and eventually when they want to shuffle the genetic deck more or less and produce the next generation, they'll go through a sexual life cycle. And that's a little pattern that we've seen over and over again. At this point, you're confronted with a horrible life cycle. Don't panic. The life cycle that they show you in this textbook is, of course, head and shoulders above anything that the BC curriculum would ask you to know. But since we're here, let's take a look at the life cycle of a cellular slime mold, where it does begin as sort of individual amoebas. The slides that follow are just snapshots of each little part. My preference is to go through the master diagram and label it. So I'll go through this and then I'll show, actually, let's pop out now and show you what the life cycle looks like before I mark that up. Now, one thing I wanted to show you, I think I have it already waiting. Let's open this in a new window. And I can zoom in on this nice little feature of Max. You can do this neat little zoom. Here we go. Here we go, nice and clear. There we go. You're looking at the life cycle, just another version of it, for the cellular version of the slime mold. And what I would focus your attention on here is the free amoeba in the center. Now, I won't get into the aspects of the diagram, other than to say there's a sexual and asexual phase, but the free amoeba are sort of the constant part of the life cycle. They're okay with that. So that's what makes them cellular. They prefer that cellular phase. Coming down to acellular slime molds, here we go, you don't see free amoebas anywhere. This is an odd sort of critter. Let's see if I can make that a little smaller. There we go. They like to hang out together in a big slimy sheath that we call a plasmodium, and they'll go through their life cycle. Sure, they'll produce spores, but they get right back to this slimy bag, this plasmodium. They don't exist as individuals, so we say that they are acellular. And I think that's what was most useful about that website. Let's minimize that now. In these animations, you're seeing a thumbnail. I find that irritating. I've asked to see if we could fix that. But down here at the bottom, if you click on Flash Video, you go to the full version. All right. This particular slime mold, and there are several genuses and several species, is a cellular slime mold. So I'm going to start the animation here and take a peek.
Amoeba-like slime mold cells live in the soil where they feed on bacteria. The free-living cells grow and reproduce by mitosis.
I want to pause them just to point out mitosis is just cloning of the existing cells, so that's asexual reproduction. They're happy to be individuals.
When food dwindles, the amoebas stream toward one another in response to a chemical signal.
That chemical signal is like chemical perfume. These cells emit a chemical which is known as cyclic AMP. That's a bit more than you really do need to know. It attracts the amoebas literally towards each other to form the larger sort of plasmodium which will ensue.
As the cells aggregate, their plasma membranes become sticky and they adhere to one another. A cellulose sheath forms around the cells. The aggregation may assume a migratory slug-like form that includes as many as 100,000 amoebas. As the slug migrates, the cells within it differentiate. When migration ends, the cells form a stalked, spore-bearing structure. When conditions are right, these spores will give rise to free-living amoebas.
Okay, so that's the end of the cycle. So when they come together, when times are tough, they'll go through a life cycle phase to regenerate the initial amoebas that you began with. There has to be a little sexual reproduction going on there, a little shuffling of the deck. This is a very good animation sitting right on the website. Just a link to a spot online. And you can locate it. It's right here. Flash video right there. Difference between cellular and acellular slime molds. I've already covered that. This one here, there we go. Just wanted to stop before it got going like a runaway freight train. Let's get back to our notes and we'll hit that life cycle. Our cellular variety of slime mold. Start. First thing to observe is an orange colored circle and a purple colored circle. The purple phase is when you get a diploid organism. At this point, and you're going to see this in a lot of our primitive sort of plants, and our slime molds are doing this trick too, where they prefer to be haploid or have one set of chromosomes. That's their preferred existence. It's only when they go through a sexual phase for a moment, produce a zygote, that they'll have the shuffling of the genetic deck, and by going through meiosis, that is the process of shuffling the cards, and you get some new amoebas emerging. Well, just understand that the whole point of fertilization is to create something new, and meiosis is picking the genes, sort of a random assortment, for the cells that it produces. Let's begin here. We've got some solitary amoeboid cells, and they seem to be pretty happy hanging out. Now they will come together when it becomes time for some kind of reproduction. So let's say that cyclic AMP smell is emitted and they all come running in, right? Like a large sort of hormonal perfume. As we come around, we've got our migrating colony, and this is when we start to see some cell differentiation, and that's neat. It's the first time we've ever really seen anything resembling differentiation of cells in this course. Bacteria didn't do that. They were just one single cell. As we go through this asexual phase, get to a point where what it wants to do is it wants to produce spores. So it needs to regenerate the amoebas. It forms a fruiting body, and spores, think of them as like lightweight little, almost like seed-like things. They're not seeds, but they're these little bodies that are dropped from almost like it looks like a mushroom cap in a way. We call it a fruiting body, and those spores regenerate the amoebas. That's a great way to do asexual reproduction. Nothing has really changed here. Our cells are all haploid. So this is a haploid life cycle. One set of genes. The reason why we say sets is when you think of conventional organisms like ourselves that are diploid, I have a set of genes from my mother and I have a set of genes from my father. So I have 2n, or 2n stands for number of sets. So I'm 2n. These little critters prefer to just have one set of genes, except when they want to go through a sexual life cycle. Let's go back to the aggregated amoebas. Now the aggregated amoebas can come together, and what we can get is we can get a diploid phase of the life cycle where we get an exchange of genetic information between some of those amoeboid cells, which we will mark up right here. So essentially, sex. Producing a 2n zygote. Now that 2n zygote can be sort of tough or resistant. Sometimes it can be a resting stage that can last through dry spells and things like that with a tough sort of outer covering. Nonetheless, when the time is right, these slime molds prefer to be haploid cells. The zygote will undergo meiosis, which takes the number of chromosomes from 2n and produces cells that are haploid, which are 1n. So all of our little cells here, our new little amoebas, are once again haploid cells. And there we go. They enter back into the life cycle, and they can go right back into the dominant haploid life cycle, where you're getting a fruiting body, which is just producing spores and asexually regenerating the amoebas. So since we said that's the haploid phase, we should say on this side that this is the diploid phase of the life cycle, or the 2n phase of the life cycle. Notice that the diploid phase is really small, and you'll see a lot of that until we move to much more advanced organisms. The remaining slides just talk about the purpose of the reproduction in general. So food becomes scarce, probably time to toughen up the population with a little bit of diversity. There is strength in numbers, and you'll find that the slime molds, they'll come together because that's the greatest chance for their survival to be ongoing. Come together as a plasmodium, go search for food, keep from drying out. You sort of get the idea. Strength in numbers. So the fruiting body, we could say, just vegetatively recycles the population. And those spores, remember, they're really quite light. As it goes up in the air, imagine if the wind catches the spores and they get scattered off. They get carried a long ways away from the parent colony, right? That's like sending your babies off to a new world. Maybe there will be food over there. Sort of seems like a smart idea, right? Even mushrooms, when they drop their conventional mushrooms, proper kingdom fungi, when they drop their spores, they're carried by the wind a heck of a long ways away, so that the babies aren't competing with the parent for food or space, things like that. Smart strategy. The acellular slime molds now have to focus in on. These are the ones that don't really hang out as free amoebas. They aggregate as quick as they can, and let's make a note here. Those cells fuse, and you get this it's sort of like the Roach Motel. Once you check in, you don't check out. If you think about each of the little cells with their nucleus, when they come together in this slimy bag, here's their nuclei like this, and they're fused. Right? There's no going back. And that is the reason why we say that they're acellular. Because a cellular existence isn't their thing. They want to merge. Interesting little structure called a plasmodium. Slimy sheath. Now here's a heck of a crazy looking life cycle for you. Again, I invite you to take a look at the colors. You look at the haploid portion of the life cycle, and you look at the diploid portion of the life cycle. Let's start with, we'll just start with our germinating babies. Begin right here. We'll simplify this. I won't test you on this life cycle, but it is worthwhile showing it to you. I have to be able to show you just enough here. Let's grab a pen tool. The cells, which have come out of they've come out of, let's see here. Where do we want to start? Let's say that they've come together and we've got a plasmodium, so a slimy sheath, and here's all their, I think this is a good place to start. They've come together here. And it's moving around as like a slug. And at this point, it's interesting because the acellular slime molds tend to go the other way. These ones this is sort of a diploid phase of their life cycle. They favor the diploid phase over the haploid phase. Interesting. And sort of suggests that moves a little bit more towards some of the other examples of life cycles you'll see in this biology course. But for now, let's say that we got a feeding plasmodium, and it goes through same kind of phase where it'll crank out a sporangium. So the plasmodium matures, and we get a sporangium, and that's something that produces these little spores. We saw that before. This side of the life cycle is diploid, which is interesting. Isn't what we expected. It's the reverse of what we saw before. Now we have a sporangium, and when these spores are produced in the sporangium, you see basically what you see going on here is a production through meiosis of these little spores. And inside of the spores, once the spores germinate, in other words, they open up and they start to grow, we get the production of the little haploid amoebas right here. These little guys are 1N cells. Those cells will come together. Acellular slime molds will experience a sexual phase. And when there's a merger of their nuclei, which is what you're seeing here, fertilization, then we get a 2N zygote. And that 2N zygote will grow up and it'll divide and it'll keep dividing, it'll keep dividing. So this is an amoeba. And it'll begin to divide and you get this large collection of amoebas. And our acellular slime molds are really uninterested in being other than a large bag of fused amoebas. They would, I guess if I drew it around here, they don't do this. Cellular slime molds would all be together like this. But acellular slime molds don't do that. They're just all together. So this is a diploid structure because this zygote is 2N. So the 2N portion of the life cycle is favored, and the 1N portion of the life cycle is really quite brief. That's the opposite of what we saw before. And it makes acellular slime molds pretty interesting. Well, I've gone through acellular versus cellular, and I think the biggest distinction you need to know about, and something I've been trying to press over and over again, is that the acellular slime molds really are not interested in being individuals, whereas the cellulars are. The cellulars prefer to be haploid, and the acellulars, they like being diploid. At this point, that would be a really good summary point for the end of this sexual life cycle and asexual life cycle. What you've got in these ensuing slides, and I'll just run through them pretty quick, or pardon me folks, I have a cold, is just a quick summary of more or less what we've already said. The production of haploid spores by meiosis going on in our sporangium right here. And they're really going light on the information here because they're saying that the haploid spores 1N, scatter and germinate into little cells that come together, and they're flagellated. I didn't mention this before, but you can see the flagella here. And those cells will come together, and you'll get the production of a 2N zygote. The only purpose of producing the spores in this case is to get them out into the wild, produce these cells for a brief time to basically have sex, and shuffle the genetic deck of the acellular slime mold. That's really it. Interesting. Water molds have been pulled into 20.5, and not that long ago, even 10 years ago, the book sources we were using with the grade 11s and with the grade 12s put the water molds in kingdom fungi proper. Now, water molds have since been reclassified as protists. Here's the reason why. Water molds don't have what it takes to be fungi. They don't have chitin in their cell walls. So that's interesting. So since they don't have that, that's one of the main things. You have to be multicellular to belong to that group. And what we think with the water molds is that what we have is just something that's just a little bit more colonial. It's pretty close to being multicellular, but the fact that it doesn't have chitinous cell walls, that's a huge detraction from it belonging to kingdom fungi. So we'll use just a simple distinction because I think that'll help you. Water molds, literally is what they say. Water molds, you find them in fish tanks. They form sort of a white, hideous mold. Let's make a note here. And these water molds aren't so much decomposers. They're actually very parasitic. They're parasitic with animals, and they're parasitic with plants. Water molds in the 1900s, they caused one of the greatest famines ever to hit Ireland. Now, this famine was known as the Irish potato famine. I have to pop out to a web browser for a second to show you this. We'll go to a few images. And there's some pretty stark images because it had such a tremendous cultural impact. People had to go overseas. People just literally left Ireland because the mold, let's see if we can, rendered the potatoes inedible. So because you couldn't consume these things, you had these horrible rotten potatoes underground. It's interesting on an iPad when you run through that. Let's see here. Here's some water molds. This is what I wanted to show. Water molds affecting, you'll see them affect the leaves. You'll see them affect the potato. It renders the potato completely inedible. And it had a dramatic social implication because although you don't realize this, maybe you hadn't heard this, potatoes are a tremendous source of nutrition. In a way, they're not a perfect food. I'd call them a near perfect food. Vitamin C, protein, carbohydrates. You can survive off potatoes for a very long time. They grow in some of the toughest conditions you can imagine, but they weren't quite ready for the water mold that attacked them in the 1900s. As you can see, you get the idea, there was a human consequence, and people in Ireland were literally starving to death and demanding that government do something about it. So water molds, let's pop out to the website. Um, when they attack animals, they'll leave. You'll see the animals embedded with the white fibers of the water mold. Let's see here. Here we go. Water molds flash video. Here we go. I'll check out for a second while this is narrated to you.
A few unrelated groups of protists are considered fungus-like because of similarities in appearance and lifestyle to the true fungi. You may have seen a water mold growing on a dead fish or insect in the water. The white cottony growth is made of fine filaments, resembling the filaments characteristic of fungi. Like fungi, most water molds obtain nutrients by decomposing dead organic matter. Other organisms in this group are parasites. An example of a parasitic species is the species that causes the disease of potato plants called late blight. That disease contributed to the Great Famine in Ireland in the 19th century. Despite their fungus-like appearance and their lack of chloroplasts, water molds are actually chromists, like diatoms and kelp.
Okay. Interesting term. They say chromists. That is another term which essentially means protist. Okay. So, unfortunately, they've introduced the term chromist, so I will correct and say protist.
The slime molds include a few distantly related groups of protists with a superficial resemblance to fungi.
This is a little bit of a review from what we just saw, but I'll let it play out.
They have complex life cycles. Plasmodial slime molds have a feeding stage in their life cycle in which the organism is a large amoeboid mass called a plasmodium. You may have seen one as a brightly colored ooze on a rock or fallen tree. Despite its size, a plasmodium is not multicellular. It's a continuous mass of cytoplasm surrounded by a single plasma membrane containing many nuclei. The plasmodium engulfs small organisms for food. It can move slowly across a surface by streaming its cytoplasm. Another group of slime molds, called cellular slime molds,
So interestingly enough, that was the acellular variety. Here comes the cellular variety.
exist as individual amoebas while food is available. When food is scarce, they come together to form a multicellular mass. This mass can assume the shape of a slug, which can crawl about in search of a new food source. Both plasmodial and cellular slime molds also resemble fungi because they develop fruiting bodies called sporangiophores, which release haploid spores that disperse into the environment. Spores that meet with favorable conditions can germinate into active cells.
There we go, nice little summary. Gives us a chance to use that as a segue, sort of finish off our notes. So let's pop back into it. Water molds start to form filaments, and that's where they start to resemble something that is definitely multicellular. Now, they don't fit into the fungal kingdom, and I know I'm sort of going over looking at whether or not they have cell walls. Having cell walls is pretty important to be in the fungal kingdom. They do that. Water molds, they don't really have cell walls made of the same material. They're a little bit out there. We've seen this before. You can reinforce your cell walls with various things. Instead of using chitin, they're using cellulose, like plants do. So it looks like they've kind of got their own little cell wall experiment going on. The differences here are so slight cell walls of chitin, cell walls of cellulose. Are water molds really multicellular? Should they be in the fungi? No, maybe they should be in the protists. This has kind of gone back and forth many times. For now, we'll just consider them to be sort of the colonial sort of protist variety. The fact that they produce motile spores, that's kind of interesting because kingdom fungi doesn't do that. The spores that are released from fungi just land on the ground and germinate. Water mold spores released into the water are flagellated. They could cruise around. That's something else that you don't see in the fungal kingdom. But remember, as we study the fungal kingdom, that's a very strange group, and we're learning more about it all the time. So here's a heck of a life cycle, but what strikes me when I look at it, first of all, is that it's heavily involved in asexual reproduction. Imagine, as we saw in the animation, that there's a dead fish floating in the water, and that's a source of nutrients. What will happen is that the water mold will begin to infect it, and what kind of shows it to you right here is they'll grow throughout that fish, and they'll sort of permeate its tissues with all the little water mold fibers, all the little filaments, and they'll take over that tissue, and they're releasing enzymes, and they'll digest it and take nutrition from it. They're saprophytes. That's what they do. Asexual reproduction is a huge element of what a water mold would do, as long as there's food. Now, water molds are pretty neat because they will go through a stage where, zoom in here, they do go through meiosis. They produce some interesting little structures here. They produce structures which will bring different cell varieties together. Technically, what you have here is something that's almost producing egg and sperm. This will come up a little bit later in the plants, but you've got sort of a male structure here. We'll call it that for now. And you've got a female structure in here. Think of it as sperm and egg coming together, because it's not far off. And this slide here shows the coupling, which is fertilization. And at that point, what we've got, there we go, we've got fertilization. So we should make a quick note that these cells at this point would be 2N. Now, that's diploid. It'll go through sexual reproduction. This thing favors the diploid portion of its life cycle, just like us. That's an advanced thing to do. It will go through asexual reproduction. It'll keep releasing little spores, like you see here. But these spores, unlike what we saw before, are diploid. So this is a life cycle where the asexual reproduction keeps reproducing basically the filaments, but what you're getting is a 2N life cycle, diploid life cycle. You get the production right here of little structures. So we'll call this the male structure. And in here, we've got a female structure. Think of it, it's almost like what's going on in animals, where you've got specialized tissues, testes producing sperm, and ovaries have produced the eggs. This is very similar to a life cycle that you would see in something like Kingdom Animalia, where they produce gametes. The gamete production really is here. That's what you're seeing in this spot here. Gamete production, gametes come together, and you get a diploid organism that maintains itself. This is a lot about the water molds. I don't go over this in class in this level of detail, but what's The reason why the water molds used to be in kingdom fungi is they had just a much more complex kind of a life cycle. So we'll leave it there. This water mold is quite a fabulous little beastie that exhibits some really advanced characteristics. What do you need to know about water molds? Well, they favor the diploid life cycle, and they're really nasty parasites of plant material, animal material that they come into contact with. How do you stop water molds? Well, I mean, once the infection cycle begins, for example, the potatoes would have to be thrown out. You just have to leave fields fallow, for example. Just take away what it is that they're eating and try to starve them off a little bit. It's just one of the many strategies. The ensuing slides, I'll invite you to go through these. This is above and beyond what would be in the course, but this slide is just showing more or less a cloning of the diploid organism. So you're getting those filaments and they're growing, and you're getting more and more filaments to the point where you're kind of getting sort of a large asexual reproduction going on. It's asexual reproduction, same as in us. If you cut your finger, the cells grow back asexually. You're diploid. There's nothing unusual here. The spores are interesting. They're flagellated. Again, you don't see flagellated spores in kingdom fungi, so this is a little bit out there. But those flagellated spores, you got to remember, this critter's in water, so those spores can find a new home. And they germinate, so that, let's switch up colors here. Let's choose a nice red. This is where they begin to germinate and grow out of the spore structure, which is right here. And that gives a chance for the filament to grow and take over its environment. And there we go. Specialized reproductive structures. The antheridia, anther is a term which means male-like. So that is probably one of the earliest structures you can imagine that's pretty similar to tissue like testes. It's producing male sex cells. The eggs would be ova, more or less. So the antheridia, just like sperm, are haploid, and the egg cells are haploid. And the last time I checked, N plus N gives you a 2N, or diploid critter. And that's how you were made. The life cycle is complete. We get the production of our little spore friends here. Fertilization gives you a diploid organism. Just remember in this life cycle that this critter prefers to be diploid. It's not suddenly going to go through meiosis as fast as it can and get back to being haploid. It's not like our slime molds. It doesn't do that. Okay, give you a chance to sort of catch up here. This is just referring to a lot more information than I would normally ask you to understand. This is the oogonium. Remember I said that there's tissues that produce egg and sperm. In a human male, the testes, they produce the sperm, and the ovaries are the place in which the eggs are produced. There's an older term for something that's that's an egg producer. We say it's an oogonium. Oo is Latin for egg. And as we talk about plant-like structures, or even fungus-like structures, where the eggs are produced, we don't say ovary tissue. That has a lot to do with animals. Instead, we say that the eggs are produced in a structure like an oogonium. So if you ever see OO, That basically means egg. And as you can see, you could earn a living talking about water molds, much less planning for how to overcome the damage that they do, because the life cycle is quite intricate. But fertilization, in here, what they're trying to tell you is that fertilization actually goes on inside of the chamber. Okay, so fertilization inside the oogonium. That's pretty advanced. So we'll leave it at that. The slides discussed it. I thought I'd go into it. The ecology of fungus-like protists, well, you'll find them in the soil. You'll find them going after matter that they can sort of recycle. They're just opportunists, more or less. If something dies, imagine if the leaf litter didn't have something to recycle it every fall. We'd have, 5-10 feet of leaf litter. So we need these guys to break things down. When organisms die, it's not just the scavengers that take care of the corpse. There's a whole lot more that works over the corpse. And, well, that's CSI for you right there. Study that. Remember that some of the fungal protists can be pretty nasty. So we've talked about the potato blight. I think I said the 1900s, so I'll correct myself. I meant the 1800s. 19th century is the 1800s, so my apologies for that mistake. But that Great Potato Famine, huge cultural ramifications, and you still hear about it today. And that takes us to our quiz. The only other thing I'd say about molds, and I thought about this at the grocery store, is just the economic severity of what molds can do. When you're growing crops and you're trying to feed the human population, and there's 7 billion people on this planet, you got to realize what the cost of fungal damage or even fungal protist damage to our food stores can be. When I was looking at strawberries tonight, I flipped the container upside down and I could see the white mold. I could see the water mold, which is the little filaments invading the strawberries and rendering them inedible. Nobody will touch it. What happens to those strawberries? They get thrown out. That's a lost sale. A farmer doesn't get paid for that. I was shopping at Save on Foods. They don't make the sale there either. So there's quite a consequence for this. We live in a temperate climate where it isn't so tremendously humid, but if you live in very humid climates, mold is a fact of life. It is a major pain. And believe it or not, they have these things called dehumidifiers, just to pull moisture out of the air to reduce the mold growth that can happen. Imagine this in your apartment. Anyways, I'll paint the picture and I'll leave it there. So folks, that was quite a bit. The water molds took up a little more of my time than I wanted, but hey, this is a recorded session, not live. So ladies and gentlemen, in the meantime and in between time, have a good one. That's it for section 20.5.